Hello everyone, welcome back to Apoptosis series. This is episode 3. I am Dr. Kalpna Rakholiya and today we are going to discuss about apoptosis versus necrosis. In last lecture we discussed about introduction of apoptosis, the discovery and history of apoptosis. In this lecture we learn about definition, morphological and molecular differences, DNA fragmentation, clinical importance of apoptosis and MCQ related this topic. So let's start. Let's learn basic things. So what is cell death? Cell death, it means cell continuously divide, function. It also is like us, and eventually cell going to die. So cell death is very important for our embryonic development, tissue remolding, removal of damaged cell, immune regulation, and cancer prevention. So in apoptosis, how cell death is occurred? So it is a very genetically program. We already discussed in last lecture. It is energy dependent, beneficial, and maintain tissue homeostasis. When we called as a necrosis, it is a pathological cell death. Is cause severely injury, uncontrolled, damage neighboring cell tissue and trigger inflammation. So this is a major difference between apoptosis and necrosis. Let's give you a simple example when we plan demolition of old building. When we are planned systematically, then surrounding area is not affected. While in case of when we accidentally demolish the building or suddenly building collapse after an earthquake, it drastically damages surrounding regions. So this is a major difference. Second, I will give another example. When our body cell inside our body cell going to normally suicide, then it apoptosis process occur. But when we are burn our skin, at that time necrosis process is occur. So let's start about difference. What is necrosis? It is an uncontrolled cell death that is caused by injury. So while in case of apoptosis, it is a programmed cell death, natural and controlled process. So here you can see that the process this is a normal cell, healthy cell with normal structure. When start the necrosis by several reasons like a heart attack, stroke, liver damage, severe infection of bacterial infection. At that time, necrosis process occur like a wound infection, abscession formation. That time, tissue going to undergoes the necrosis. So this is normal cell. This normal cell going to swell, then membrane going to rupture and become it. This rupture cell affect the nearby other tissue as well as the nearby cells, and it goes to inflammate. It inflammate the in surrounding tissue. While in case of apoptosis, you can see here this is normal cell. When this cell are going to apoptosis process in during the development of embryogenesis, menstruation, shedding of the uterine lining due to apoptosis endothermal cell, immune system regulations, skin cell turnover, during that cell going to cell shrinkage first of all. Second is a formation chromatin condensation and formation of apoptic small apoptic body. This apoptic body is with the membrane bound, so there is no chances of cellular cytoplasmic leakage. And once this small apoptic body is formation, next macrophage are going to activate. This macrophage going to uh, engulf this apoptic body, so there is a no inflammation occur. This is the main difference between necrosis and apoptosis process. So these are the various uh, examples in which you can see that another example of necrosis: trauma, burns, toxin, eczema, infection, hypoxia. But in this apoptosis condition, it require ATP dependent process. While here you can see there here in case of this is also ATP dependent, but here cell loses ATP as well as ion pumps going to fail. Ion, it means sodium potassium ion channel is going to fail. Water enter in the cell going to swell. Finally, membrane are ruptured. Intracellular content, whatever is present, it will leak out without the cell membrane, and this leakage activate the immune cell, and inflammation is occur. So this is the cell gradually shrink, cytoplasma become dense, chromatin dense, nucleus fragmentation occurs. As we discussed, fragmentation occurs between specific base pair. Precisely, it will fragmented. Plasma membrane remain intact. Small membrane body, apoptic body is formed. Macrophage quickly engulf the and no inflammation occur. This is the occur in morphological changes occur in apoptosis, while in case of necrosis, it is a totally opposite to the apoptosis. Cell going to swell, organelles going to swell, mitochondria become dysfunctional, lysosome going to rupture, plasma membrane breaks, then cell contents spill out outside. Inflammation begin immediately, and neighboring cell may also be damaged. Molecular mechanism. When we talk about the molecular mechanism, a major molecular event occur when cell going to suicide. First, death signal is arise. Then after initiator caspase going to activate. They are normally in a procaspase form, so it is in form of inactive. Then after this signaling, it initiate the caspase enzyme. Then executor caspase are involved, then protein are going to degrade, DNA fragmentation occur, apoptotic body formation, and then phagocytosis clear this kind of cell. In case of necrosis, no caspase activation. Instead of ATP is going to depleted, loss of ion like sodium, potassium, and calcium. Calcium ion are overloaded, as well as reactive oxygen species are generated, and drastically membrane are going to rupture. There is a once these are cytoplasma are leak out into the surrounding region, more immune cell are activate, and inflammation will arise. When we are talking about the difference in DNA fragmentation, in case of we already discussed in last lecture, in apoptosis process, DNA is cut between nucleosome. It is a very precisely manner in a very precise base pair structure, like it produce a fragment between A180 to 200 base pair, and it give the when we load the in a gel electrophoresis, it give the DNA ladder like a pattern. This is a one of the hallmark for the apoptosis process. While in case of the necrosis process, DNA is not a degrade in a precise form, it degrade randomly, and that's why it produce when we run this necrosis DNA into the gel, it produces a smear like a structure, blur like structure, not a give a distinct fragment in a gel electrophoresis. This is the most important topic. It will ask in a, CSIR-NET exam. What is the difference main between apoptosis, DNA fragmentation, and necrosis? So in necrosis case, is the appear smear like a structure in the gel electrophoresis, while here it give the DNA ladder like a pattern. So remember, this is the clinical importance, how excessive apoptosis process is required in a homeostasis process. It means it should be balanced, not in a excess apoptosis process required, and not a reduced form of apoptosis. If excessive apoptosis occur, then it lead to Alzheimer's disease, Parkinson, Huntington disease, AIDS, ischemic injury. So it means excessive loss of cell, impaired tissue and organ function. If a reduced apoptosis is apoptosis process going to slow or not happen at that time, cancer, autoimmune disorders, viral persistence, these are the occur. So how reduce apoptosis occur? Due to the one kind of a marker, that is BCL2, that is mutation in BCL2. BCL2 it involve this kind of protein involved in apoptosis process. So if a mutation in BCL2 cells survive for a longer period of time, and cancer will be developed. So, reduced apoptosis allow abnormal cell to survive and accumulate. While in case of necrosis, it will common seen in a heart attack, stroke, burn, gangrene. Gas gangrene is a one kind of lethal disease that is caused by Clostridium perfringens organism. that is gram negative bacteria, which will survive in anaerobic condition. In that case, necrosis process occurs. This microorganism grow inside the tissue, damage the tissue, and it is a very painful process. And sometimes people are going to ampule their leg, whatever part is affected. So that is there is a no cure for that process. During the severe bacterial infection, necrosis processes occur. So there is a clinical importance. These are the very important MCQ. You have to take the screenshot and give me the answer. I will give you the correct the answer keys. Thank you very much for listening this episode.